Okay, uh, today we are going to discuss the data response question of this is from May. Today we are going to discuss the data response question from IB paper May 2018 time zone 2. Now this question is combination of topic 9, which is plants, topic 9 plants, and it has gene expression part, which is topic 3 genetics. Now if look at the question. Arabidopsis is a small flowering plant in the mustard family. The advantage of using this plant is it's having a short life cycle and it flowers quickly, producing a large number of seeds and is easy to cultivate. It forms a circle of leaves known as rosette here that lies close to the soil. Flowers form at the end of short stems, so flowers are forming here. Now there are 4 stages of this plant. Now the study conducted on this plant by growing it in a long day. 16 hours light is given in a long day, and same plant was grown in a short day as well, where 8 hours light is given. So the same plant was studied. Plant is a control variable, but the length of the day, the light, is the variable which we are going to study. Now the meristem grow by the meristem and go through 4 stages where we study. S1 rapid cell division, S2 cell division ceased but cell expansion continue, S3 cell expansion continues but the rate decreases, and S4 is for the leaf growth completion. Now there is a graph given of the mean leaf area. The study was done on leaf number 6. As we know, the rosette is formed, and once the 6th leaf came, we study it. The mean leaf area was studied and mean number of leaves in the rosette is studied. Now the graph, you can see that it's from day 0 to day 30, and length is going from 0 to 150 leaf area, number of leaves in that 30 days 1 month is going from 0 to 35 in the graph. Now look at the question. The question 1 is asking about to calculate the difference in the mean leaf area at stage 4 between the leaves of plant in long day and short day. So we have to look at the growth in stage 4. Here I use line. So stage 4 is this one, is somewhere here, and for this one it's somewhere here. All right, so this one I will say around 135, and this one will be 50, 80 or 85. Let's take 80 or 85, it's depend. 100, 10, 20, or this one 130 or 180. So 1 30 minus 80, so 50 mm square. In the mark scheme it is given 45, but the range of 40 to 50 is accepted as the uncertainty. So this is what is given. Let me double check. 50, 60, 70, 80, little above than 80 probably my line, but this is accepted. But in this question, if you do not put the unit, you will not get full, means 1 mark. So unit is must for this question. So this is around 50 mm square. Now distinguish between plants grown in long day and short day in the timing of 4 stages. Now you have to see all the 4 stages, and distinguish means you are going to not give similarities, it is only you have to give differences between plant grown in long day, short day in all the 4 stages. So if I look at these stages, I will look at this part. You can look here as well, but I will focus on this part. So I will say S1, S2 is longer. You can see S1 and S2 longer in short day plant, while shorter in long day plant. So S1, S2 longer in short day plant, while shorter in long day plant. Now my focus is here, it is 2 mark, I have given 1 point here. Go back and have a look again. So probably I can say S1, S2, S3 phases in long day plant complete earlier, right? Or it's very short. S1, S2 and S3 completed earlier in plant grown in long day. S1, S2, S3 completed earlier in plants grown in long days, while I have to complete the sentence. One thing I'm making mistake here, my answer should not go out of the box, but because it's like, you know, I'm typing on iPad, but you need to make sure that your answer should be within the box. While S1, S2, S3 and S4 have while in short day it takes longer to reach S3, right? Another thing you can look here, many other points, 2 points I have given, but just to tell you, this S1 in long day plants, S1 stage reach later in long day plant than short day. So this point also you can mention there. So these are the points which you can, and you can see the length of all these stages is also different. In long day plant there is more variation in all the stages in long day plant. So any points you could have discussed, you can look it from here, from the graph or from the top. S1 stage reached earlier. You can see S1 stage, S4 sorry, S4 stage reached earlier in long day plant compare to short day plant. Short day plant we are reaching S4 here. okay, around day 30, and here we are reaching here so earlier. So these all points are for distinguish. You can talk about any of these points. Next question, between plants grown in long day and short day, in the mean number of leaves per rosette. It means now we have to look at this graph. Mean number of leaves per rosette. We can see here in short day plant the number of leaves are much more higher than the long day plant. So that is our 1 point. I just write down here. So you can simply write down rosette of plant grown in long day has fewer leaves than short day. Okay. Another thing, number of leaves, and then another thing you can see that it become constant in long day plant. It become constant after around 10 days. So this you can say that leaf number in long day become constant or plateaus or stay constant, while in short day number continues to increase. So that is the difference you can see. I'm answering this question here so that you can, it's easy for you to see the, so this is part 1C, that 2 points. You can talk about these 2 points here. Now look at the next question. Leaves were removed from Arabidopsis plant that had been grown in long day and short day, and the concentration of starch been measured. Now here this was done at the end of the day and night. Alright, so all 4 stages are taken and the concentration of starch being checked. So our independent variable is day and night, while our dependent is starch concentration, and 2 variables grown in short days and long day. Discuss the evidence. Now we have a command term discuss, which gives us the liberty to to prove or to support the hypothesis or to reject the hypothesis. So let's look at the hypothesis that the plant leaves use up starch reserve for cell respiration during the night. Okay, so we have to look at the evidence that whether the plant leaves are using up the starch. So we have to focus at this part: night, 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 and night. Now we see that definitely during night time the starch levels are low. So leaves used up the starch, leaves used up the starch at night as the level of starch is lower at end of night in all 4 stages, right? Comparatively, if you compare it with the daytime. So this is one of the evidence. Yes, the starch concentration is low. But one thing, it is again 2 points. We have to give 2 points. So, but another point which is you can add, there is no evidence, because the question is saying the starch reserve for cell respiration. So this also you have to put as a point. No evidence that starch is being used for respiration. It is nowhere in the graph shown that it is used up as a respiration. So if, let's say, if I want it to be as a point to put that it is used up for respiration or not, I probably can have one more variable here, cell respiration, and then I can show my cell respiration activity here. Rate I can include, but because this is not included here, so I'm not sure that this is because of cell respiration or for other activities. So because it is saying you to discuss, you can put this point, no evidence that a starch is being used for respiration, or starch can be used for other things. All right, so this is how we work with discuss. For each of stages, identify whether the starch concentration at end of day is higher in the leaves grown in long day or short day. So if you see starch concentration is higher in stage 1, so I probably answer here 1F, higher for S1 in short day plant. Let's look at short day first. Higher one, but it's lower in S2, S3 and S4 phase for all, right? S1 higher, higher in plant grown in short day in S1, and high in plants grown in long day for all stages. Other it's lower, but for all higher in long day. So you can phrase it this way. It's 1 mark. Suggest reason for the difference in end of day starch concentration in S2. So let's look at S2. S2 starch concentration in long day and short day for both. Difference in S2 for plant in long day and short day. So we focus on short day, and the concentration is more, starch concentration is more in long day. I write down starch concentration more in long day while it is lesser than the in short day, right, in S2 phase. Okay, now are they asking you about the day or night thing? Anything about day night? End of day. So we don't look at the night, we only look at this part. Now what is the difference? Now you can add your theory, knowledge of theory, that leaves in plant grown in long day receive more light. Being long day, plant receive more light for photosynthesis, more starch concentration, thereby more starch concentration. You can see that. Okay, or you can attach with the information of leaf area here. You can see the leaf area is more, so more leaf area more concentration. Okay, now in short day plant, the starch is more for production of more leaves. You can see that they are producing more leaf, number of leaves is more. So in S2 phase, and you see that they have a long S2 phase here. Here this is S2 phase, and they keep on making leaves. So starch is getting used up to make leaves. So I'm putting the second point here. In short day, right, plant using starch for producing more leaves. Okay, So that's the difference. In the long day, they are getting more sunlight. They are photosynthesizing more, and they are not using that the food. They are not making many leaves. The number of leaves are not many. You can see this, it become constant. So it means they can store more starch. While in short day plant, because being the day is short, the rate of photosynthesis is less. Why? And on the other hand, they are using it for making more leaves. That's why the starch concentration is low. Look at the next part of this question. To account for observed phenotypic metabolic differences, research analyzed the messenger RNA transcribed data, and they found some data was overrepresented. Some data is overrepresented during the long day. Compare with that during short days. So you can see few things are changed here and here. Using the data and discuss the evidence for plant adapting to different daylight regime by changing the pattern of gene expression. So you can see, and you can connect it with your information, general information, that long days means more sunlight. More sunlight means they can have more metabolism. All right, so this all is oxidation reduction reaction, which is the part of your Calvin cycle, and metabolism of nucleotide. Calcium signaling is to take more water from the soil. So all these things, because it's more sunlight, more photosynthesis is happening, so these all parts are expressed so that it can facilitate with the, can adapt the plant for the high rate of sunlight. Now short days, being short day, less sunlight. If there is less sunlight, means they have to adapt in a way that they should utilize the amount of sunlight given to them to the maximum. To the maximum means they must have more photosystem. More photosystem means light dependent reaction. Light dependent reaction rate should increase. Phototropic response, they should respond to the limited amount of light to get it maximum. Sugar transport, if the transport is more, if the genes are expressed in a way that the transport of sugar is fast, then they can quickly make more food. RNA processing, amino acid transport, all these are the part of the adaptation for the limited amount of light provided to the plant. All right, so you can provide any one of these example from the bar graph given to you. First of all, we can say transcript factors. They definitely are different. You can see none of them are overlapping or the same. So transcripts differ in plant grown in long day and short day. So this is very clearly, you can see the amino acid transport, transcription is in short day, while in long day you can take anyone, oxidation reduction reaction, or you can say the secondary metabolism transcription occur. Okay, so 1 point. Now what else is there? Different genes are being expressed. It's clearly indicating different gene expression indicated by RNA processing. You can give any in short day, any valid point you can give. Third point you can give, plant adapted, any example, plant adapted at different daylight by altering gene expression. So produce more leaves in short day, or larger leaf in long day. So if there are larger leaves in long day, which may result in more transcript for biotic extracts or more transcript for calcium signaling, any one thing you can put as third mark. Now last part, using all relevant data. Using all relevant data in this question, deduce with reason whether Arabidopsis is a long day or short day plant. Now I will go through with all the graph and we see whether it is a long day or short day. So for long day, the flowering should occur during the daytime, means during the longer period of day. So here if you see, it is flowering hormone metabolism is happening here. So the metabolism is happening for flowering during the long day. So it's a long day plant. This is the evidence from this long day plant. We'll look at the graph as well. Long day plant. First evidence I can give from this graph, flowering hormone metabolism gene over represented in long day, right? Looking at this graph, if I see the number of leaves, because we know that the flowering is happening here on the top and the leaves are there. So if you see, the number of leaves are more in the short day and less in the long day. Why is it so? Because during long day, the metabolic process of flowering is happening, which is not letting more leaves to produce. You understand? So during long day, there are fewer leaves in long day as the energy shifted to the process of flowering. Okay, or you can say that more leaves in short day as they don't need energy for flowering. So any vice versa, you can use a valid reason for your data, and that's all as your